Thank you, Madam Chair and committee members. As stated, my name is Rick Harrison. I've been a pest control advisor for 16 years in Ventura County. I also serve the California Association of Pest Control Advisors on their executive board as the treasurer. My roles as a PCA consist of field checking. I walk orchards and monitor pest populations and beneficial insect populations. I trap for pests such as Asian citrus psyllid, and I monitor the edges of fields primarily. There's a edge effect, what they call an edge effect with Asian citrus psyllid, where you see a lot of the pest, the beginning of the pest population forming at the edges of fields. So that's the first place that I look. And also when the trees are flushing with new growth, they are very persistent insects, and they will find the smallest leaf that you can imagine and lay eggs on that. The problem with Asian citrus psyllid is it reproduces very fast. It can go from an egg to an adult in as little as 2.5 weeks. And each adult can lay up to 800 eggs. In our area in Ventura County, we have started with the area-wide treatments, where we're basically working from the east end of our county and spraying every PMA, which is a pest management area. Each location needs to be sprayed within a 2-week period. And then after that 2-week period and the people have, commercial citrus has been treated, CDFA will come in and treat the homeowners around there. Right now we're just finishing our winter treatments. We have approximately 2 weeks left on that. And in areas where we began the winter treatments in January, I'm already on my 3rd spray, because the pest comes back so fast. The pesticides that we have available are very effective. However, they don't last long. Of the pesticides that we use, the pyrethroids and the neonicotinoids seem to be the best. The problem with these chemicals is if we're continually spraying the same materials over and over to control the pest, eventually resistance to the pesticides can happen, and I believe they're starting to see a little bit of that in Florida now. These pyrethroid sprays do upset our natural enemies, so when we do a spray, we're creating other problems. We see other pests come up that we normally don't even have to treat for. So these treatments that we're putting on are a cost to the grower, and then we're also creating another treatment down the line that's another cost for the grower. So eventually, it's my fear that we're going to get the growers to not want to treat anymore because they feel like they won't be able to afford it.
So are you saying that the treatment in itself reduces the resistance to yet another disease?
So basically what happens is in Ventura County, we have a very unique system where it's a coastal community, the weather is very nice, and we have a lot of beneficial insects that control other pests such as spider mites and red scale. And then when we go and spray these chemicals for the psyllid, we're knocking those beneficial insects out, and you have flare-ups basically of the other pests.
I see. Thank you.
So I also deal with organic citrus, and there are organic pesticides available to use. Many of them are very short-lived. The 2 best materials that I've seen don't really control the insect for more than a week, if that, if you're lucky. So I don't think or I would hope that organic would stay around, but it's not looking so positive.
The organic treatments?
Yeah.
Okay.
Something that has been difficult for us is getting releases of parasitoids. Like we heard earlier, Tamarixia is a very effective parasitoid for the larval stages of Asian citrus psyllid. The biggest concern for me is we are not allowed to have private industry rear the insect, and I don't know if CDFA has the capability to keep up with the demand that's necessary. We have a lot of homeowners that are very close to citrus, commercial citrus, and in my mind, that would be the ideal situation for Tamarixia releases, is to release in homeowners citrus. Something that I deal with CAPCA is educating PCAs, facilitating the success of the PCA through education. And in order for me to maintain my license, I have to have 40 hours of continuing education that is, I guess, certified through the Department of Pesticide Regulations. And it's becoming difficult for a lot of the courses to be approved by Department of Pesticide Regulations recently. And I think my last thing is, in order to reduce the spread of the disease, we need to reduce the population of psyllids and prevent the movement of infected psyllids into our area or any growing area.
Okay. So the Camarixias are not able to be reared by the private sector at all, so there's just a reliance on CDFA for those bugs?
That's correct.
So currently, the biocontrol program, there's CDFA. We have a rearing facility at Mount Rubidoux. We're in the process of almost having one built at Cal Poly Pomona. USDA does some rearing, and they're doing what we call field insectaries, where they actually tent trees and rear the Tamarixia in the field in these tents. They also have partnered with, under USDA, a private insectary in Southern California to develop what we're calling the technology transfer. The idea is that once this is developed, and I think we're very close to that, that technology can be transferred to other private insectaries. And we're hopeful that we can get them to where they need to be to start rearing Tamarixia. It is a very labor-intensive insect to rear. Very different from many other biocontrol agents. You require nursery plants, you require a colony of ACP, and you require Tamarixia to continually rear the Tamarixia. But that is something that's in our short-term plan, is to get this technology transferred over to any insectary, private insectary that's willing and able and interested, and then help facilitate the permitting process for them to start rearing Tamarixia. Because we do see that the goal of our program is really the urban residential area, and that private insectaries could fill the niche for growers.
Okay, thank you. Any other questions? Okay, thank you very, very much. And we are moving into our last panel, Developing Solutions to Combat Asian Citrus Psyllid, Huang Long Bing. And first we have Beth Grafton Cardwell, PhD, Director of Lynn Cove Research and Extension Center and Research Entomologist at UC Riverside. And following the presentation from Beth Grafton Cardwell, we have Neil McRoberts, PhD, and Associate Professor of Plant Pathology at UC Davis. Thank you.
Thank you, Madam Chairman and the committee. Thank you for inviting me today to speak about this. I did provide a PowerPoint handout for you if you want to follow along with that. That might help with the discussion. My name's Dr. Beth Grafton-Cardwell. I'm a citrus integrated pest management specialist hired by the University of California, Riverside, but stationed up in the San Joaquin Valley, where for the past 26 years I've been studying citrus pests and how to manage them in the best possible way. I am also director of the Lynn Cove Research and Extension Center in Tulare County, and that particular center has two roles. It provides a place for researchers to come and conduct research on citrus trees, and it also houses the Citrus Clonal Protection Program, which is a screened-in area of trees that they release budwood that has been tested for diseases, and they release that to the nurseries who then make the trees that go out to the commercial growers as well as the homeowners. So it's a way for the citrus industry to ensure that people start with disease-free trees. And so it's a very important aspect of that particular center. Now, I'm going to go backwards a little bit into some details of the biology of the psyllid and huanglongbing, because I think it helps explain why we have the research directions that we have and why this is a really difficult research problem. There are two ways that huanglongbing disease can spread. It can spread by grafting diseased plant material from one tree to another, or by the Asian citrus psyllid. The Asian citrus psyllid picks up the bacterium when it's feeding into its body. It recirculates and increases, and then when it feeds on other trees, sometimes, not always, but sometimes it passes that bacteria into the next tree and passes it on. And HLB is a deadly disease. It causes the fruit to become small, uneven in size and color, and it renders the juice completely unpalatable. It becomes bitter, and it can't be marketed. And then as the disease progresses in the tree, it eventually kills the tree, and currently we have no cure for that disease. Trees can die in as little as 5 years. We expect citrus trees to live 100 years, so it's very shocking for us to have a disease in North America that can kill trees that quickly. We estimate there are about 30 million commercial citrus trees and probably 20 million residential trees in California, so the disease is affecting everyone. And as it progresses through the state of California, it's going to change the landscape of California because we have so many trees that are citrus. The disease was first discovered in Florida in 2005, and it swept throughout that state, largely because Florida did not do anything to manage the psyllids. Now it's estimated that 100% of commercial citrus orchards in Florida have some level of the infection. And as you heard from the Floridian, that acreage has declined by 50%, production is down more than 60%, and their industry is being threatened to collapse because there isn't enough fruit to sustain the juice plants. California first discovered the disease in 2012, but has managed to confine its spread to Southern California for the moment. California is doing better at controlling huanglongbing compared to Florida because it proactively instituted quarantines and regulations to help prevent spread of the disease. The citrus nurseries have moved a large part of their production inside of protective structures, either screen houses or greenhouses, and they're treating plants with pesticides before they ship them out to the retail markets. And all this is to ensure that the trees are protected as they're sitting out there in the open so that psyllids don't come in and establish on them and then get bought by homeowners and moved around the state. Plant material in the form of trees or bulk citrus cannot move freely out of the quarantine areas, which in this particular picture on number 5 is shown in blue are the quarantine areas. So presumably where you have infestations of psyllids, you don't want bulk citrus that hasn't been treated or nursery trees that haven't been treated moving out of there and carrying psyllids with them. Commercial citrus orchards are being treated with insecticides. Residences are being treated with insecticides if they're in a zone that is new to the insect or near commercial citrus. And that's in an effort to locally eradicate the pest, especially in those new regions. In areas where the psyllid has become well established, such as Southern California, parasite releases, you heard about the little Tamarixia, are being made, and homeowners are being asked to treat on their own for ants to protect those parasites and apply insecticides when they can to help control the psyllids. But it's a really difficult problem. As you all know, it's very difficult to get the urban population to control pests, and they have a hard time understanding what this insect is. It's as small as an aphid. Most of them barely know what an aphid is, much less what a psyllid is. I've spent my whole life studying the insecticides that control insects in citrus, and many insecticide groups kill psyllids if you make a direct hit on that particular psyllid. However, the psyllids are extremely tiny and they move about, and it's difficult to achieve perfect coverage with any spray. Therefore, it's important that insecticides with lasting residues are used so that the adults that fly in from a neighboring field or the nymph that emerges from inside its little nymphal case and comes out as an adult and somehow has escaped that direct hit, they get hit by the residues that are still on the leaves. Research worldwide is showing that the neonicotinoid group and the pyrethroid groups are absolutely the longest lasting in terms of their effect on psyllids, and that's why they're used quite a bit. They're used in both the residential treatment program and in commercial growers as well use them quite a bit. So maintaining registration of these insecticides is absolutely critical for management of psyllids in both backyards and commercial citrus. Most citrus varieties bloom for only about 4 weeks out of the year, so bee health can be easily preserved by careful timing and use of these insecticides. And regulations are in effect to protect commercial citrus, and CDFA is very careful not to spray these insecticides on blooming trees in backyards. So a lot of the concern about pesticides related to bee health is really, people are not very well educated in how the products are used and how they can be used safely and protect bees. So I think that's an important point to make.
Yes.
Because of my expertise in pest management of citrus, much of my time is spent developing the treatment guidelines for citrus growers and homeowners for how to respond to the Asian citrus psyllid and Huanglongbing disease. And these guidelines are available from University of California Ag and Natural Resources website and UCIPM, University of California Integrated Pest Management websites on the links I've shown in my handout if you want some more information. Now let's get to the disease. Why is this disease so rapidly spreading and so scary? This disease spreads extremely fast because when the psyllid lays its egg on the leaf, it also feeds, and it may deposit the bacteria right next to where it lays the egg. So when the egg hatches and the nymph emerges, it's feeding on a localized bacterial infection and picking it up immediately. And it retains that bacteria in its body when it molts and becomes an adult and flies away, and it's carrying that disease for 6 weeks. Now, we can't tell if that tree is infected or not immediately because when we go up to test it, we grab a leaf from the tree, and that may not be where that localized bacterial infection is. It may take a year for that bacteria to circulate through the tree to the point where when we pull a leaf and test it with a biochemical test, we go, oh yeah, the bacteria is there. And so during that time that we can't detect the disease, the psyllids are already spreading it to additional trees. So when we say we have 21 trees that are infected in San Gabriel, there's probably a lot more than that that are infected, but they're in the early stages of infection and we can't detect it yet. Ultimately, all this spraying for psyllids is just a stopgap, and ultimately what we need is a cure for the disease. Suppressing the psyllid with insecticides is extremely difficult because it's small, there's a lot of them, we can't really tell where they are. And it doesn't stop disease spread because it only takes a few psyllids to spread the disease. And that's why biological control techniques and organic insecticides are weak, because they're not killing all of the psyllids. And if it only takes a few to spread the disease, it just keeps spreading. At best, psyllid control is just buying time for researchers to develop solutions to the disease problem. And there are literally hundreds of scientists around the world looking at every conceivable way to control this particular disease. They're looking at altering the plant through both traditional breeding methods and genetic engineering. so that it can withstand the bacteria. They might be using spinach genes that carry an anti-HLB gene on them into the plant. They may be using traditional methods such as, okay, in Florida, we have all these varieties of citrus, and these two varieties seem to be doing a little bit better than the rest of them. Let's encourage those varieties and look at the genetics of those varieties, and maybe we can make a super citrus plant. They are using a genetically engineered citrus virus to act like an inoculation and carry anti-HLB genes into citrus trees. That's another method they're looking at. They're attempting to alter the psyllid so that it can't pick up the bacterium. And the thought with that would be they would release this new psyllid into the environment, and it would outcompete the wild psyllids, and it would reduce the amount of bacteria in the environment. They're looking at the use of antibiotics to reduce bacteria in the plant. They're looking at odors that attract the psyllids and could maybe attract them to a trap that would kill them. And the fact sheet that you have in your particular, in the front of your set of handouts, talks about all of the University of California research and extension programs that are going on right now, paying attention to this pest and disease. In the end, it's likely to take multiple tactics and quite probably genetic engineering to stop HLB from decimating citrus, California citrus. However, a lot of these tactics that are being looked at are still 5 to 10 years out. And you've heard that in less than 10 years, 100% of Florida citrus was infected with the disease. So we don't have a lot of time. We have to work very, very fast. There are millions of dollars in federal funding committed to fight HLB, and more are needed to address the disease before it gets a foothold in California. One of the most important research areas for California is to develop and validate what we call early detection techniques that will find the bacterium soon after the tree becomes infected. EDTs may use dogs or machines that kind of sniff the profile of the tree and say, oh, it's changed, it's not healthy anymore. Or they may grind up plant material and look for proteins that the bacteria produces, or chemicals that the tree produces, because it says, I'm being affected by this disease and I'm putting up an immune response. These sorts of responses move throughout the tree fairly quickly, so instead of finding a tree that's infected with a biochemical test after 9 or 12 months, we might be able to find it's infected after 2 or 3 months and kind of get ahead of the disease spread. So I think that funding is critically needed for these early detection techniques to bring them to fruition before the disease spreads any further. These techniques could be used by growers or homeowners to detect which trees are early in their infection and get rid of them, and so slow the rate of the disease spread. Remember, Florida and Texas did not have these tactics in place when the disease first appeared. They couldn't detect the disease and remove the sick trees fast enough, and now they have high levels of infection. California has an opportunity to stay ahead of the disease. And then I'll just finish with a great resource for keeping abreast of the research activities going on in California and beyond is Citrograph magazine. It's produced by the Citrus Research Board, and many of the articles are written by grower-funded researchers, and it's written for California growers. I'll finish by saying that California is in a much better situation than Florida, who did nothing up front, because of the regulations restricting pest and plant movement, the huge commitment of resources by the California citrus growers, and the amazing cutting-edge research that's taking place in California. Anything that the Senate Ag Committee can do to further these activities is very much appreciated. Thank you.
Thank you very much. Any questions? Okay. And next we have Neil McRoberts, PhD, Associate Professor of Plant Pathology from UC Davis. Thank you.
Good morning, Madam Chairman. Thank you for inviting me here. Thank you to the members of the committee to be here and hear the evidence. So I'm going to try and act as clean-up hitter here and pick up on a few points that other speakers have mentioned and pick up on a couple of things in particular that Beth covered. I'm a plant disease epidemiologist. I joined the faculty in Davis in 2010. Before that, I was in Edinburgh in Scotland, where I ran a research program that was focused on sustainability research for the Scottish Government, a lot of policy related research. The work that I do on HLB, I started in 2012, collaborating with the Citrus Research Board here in California and with Beth, and we work closely together in developing a lot of the kind of practical programs that are being put in place to try and combat this disease. And as much as I'm a plant disease epidemiologist and obviously professionally fascinated by the bacteria itself, I would say that I think the major components in stopping the disease spread here in California are not anything to do with the insect or the bacterium itself, but actually there are human elements that dominate the dynamics of the disease. And we've heard a lot about them this morning. And I think there's a great deal that the state can do to enhance the control efforts that are going on. For example, at the moment, because the citrus that's grown in commercial orchards is all produced from certified material, the probability of there being infected trees in groves is very, very low. And the major sources of inoculum, the major sources of infected trees are likely to be in urban areas. And we know that there are infected trees in Hacienda Heights and San Gabriel. That bears out that idea. And so it's a difficult job to actually keep all of the growers focused on the idea that they're fighting a disease which isn't actually there at the moment. They have day-to-day concerns. The things they were already concerned about didn't go away when HLB arrived in the state. They have to think about all of those things already. So as much as it's true that at an industry level, the industry is very, very committed to help itself to fight this problem, at the ground level, with thousands of individual growers, it's very difficult to keep all of them focused on the problem that this disease poses for the industry. And so you can never have too much extension when you're facing a problem like this. Extension is expensive, but boots on the ground go a long way to getting growers organized. And ultimately, as you heard from Florida, it's the local organization of growers to do the right thing at the right time and to stay together, to act as groups, to behave as good neighbors to one another. Those things will actually achieve some success. But there's a long lead-in time. In an ideal world, we'd never get there, but there's a long lead-in time to a lot of those guys actually having to do things to combat the disease. And so the network of grower facilitators who work with the growers is fairly thinly stretched. There aren't a large number of those people who have to keep the growers focused on the problem. Bob Atkins and his team do a fantastic job, but more boots on the ground would make the job easier and it would make it more effective, and it would significantly increase the chances of the psyllid management area program succeeding in its objectives. So there are cooperation problems, coordination problems at a local grower level that more resources would help with. We've heard a lot also today about the interaction between the different agencies involved in trying to combat the disease. And again, while it's true that the interactions are fantastically successful, more can be done to help ease some of the problems there are with those interactions. Each of the institutions involved has its own constraints, its own objectives, and those have to be compromised to some extent to achieve success. For example, the status of a tree as positive or negative for HLB is a legally defined definition that is dependent on a particular diagnostic test result. It has to be that way in order for there to be a legal basis for the regulatory agency to be able to take action and for example, issue a warrant to say that an infected tree can be removed. Infected has to have a legal definition. Unfortunately, for the growers, there's often a long delay, as Beth just explained, between when a tree becomes initially infected and when that legally defined definition of infected will be reached. And so there's a certain amount of tension between the growers and the regulatory agencies. It's natural for both that this should occur, because the growers would like the trees to be detected faster and removed quicker from the urban area than it's currently possible to do. But it would be very difficult to establish a legal basis for the early detection technologies to say for definite that a tree was infected. And of course, that gets us into very difficult water then when it comes to trying to force individuals to have trees removed from their properties. And so there are some complex issues that need further analysis and further work, and a lot of facilitation as we work towards trying to get rid of the HLB from the system. I have to say, as an epidemiologist, I believe that objective is actually possible in California. I believe we're not yet past the point where we could stop the progress of the disease in its tracks, but it will require even better collaboration and cooperation in future between the agencies involved. And at a fairly high level, some direct intervention to ease the wheels of collaboration between the private sector and the regulatory agencies would go a long way to help with that. One example where extra resources for enforcement would be effective is in the area of the issue of fruit movement that we heard about from the director of the Cecilia Packing Company. As he explained very eloquently, those regulations to require growers to clean and tarp loads before they're moved between areas are costly, but they're also very, very necessary to help slow down the spread of disease. So on the one hand, the industry can see that those things are beneficial, but on the other hand, they impose unwelcome costs and constraints on the industry. Currently, if you spend some time in the San Joaquin Valley, it's not that difficult to see untarped truckloads of fruit being moved around, essentially because there's very little, if any, enforcement of those regulations. There simply aren't resources to enforce those regulations, and human nature being what it is, human beings cut corners. And so some extra regulations for enforcement, some extra resources for enforcement would go a long way to helping with cut down those kinds of opportunities for human agencies to move the problem around. Beth Stone-Smith and others got into talking a little bit about the dynamics of the psyllids and how much bacteria they carry and where they are in relation to the infection. Dr. David Bartels, who works for APHIS in Texas, is actually coordinating studies where he takes all of the psyllid sample results and maps them out. And he has some very good data showing where there are clusters of psyllids that have suspect levels of results from PCR tests. And either Beth or myself could put you in touch with Dr. Bartels if the committee wants to see that data. It's available in a ready, digestible form, and Dr. Bartels is carrying on doing those analyses. And that data, which shows us where those suspect psyllids are, is being used by CDFA and by USDA ARS to evaluate the risks and to help coordinate the survey program. But that information will let you see exactly what portion of the psyllid population have the bacteria and what portion don't. And the story that happened in Florida was that initially very few of the psyllids have much bacteria in them because there are few infected trees for them to get it from. But as the proportion of infected psyllids increases, more trees become infected, and we get into a kind of feedback system where eventually a very large proportion of the psyllids end up with the bacterium in them, and the disease spreads in an uncontrolled way. We're not yet there in California, and I believe the opportunity still exists to stop us from getting there. But again, action is required immediately to do all of the things that need to be done to prevent it from happening. There's some, just again, picking up on the idea of tagging trees, which was mentioned, and the relatively low level of effectiveness of tagging trees. A research proposal has just been submitted to the Citrus Research Board to look at that issue, to look at tag design, and also to help maybe to gather information from the tags using smartphone technology to actually look at where plants are moving to, who's buying them, and those sorts of issues. So there is some work in prospect. Again, it would be industry funded to have a look at the efficacy of the tagging process to try and prevent the spread of disease. I have to say that a lot of the problems to do with urban spread of the disease are connected with some fairly prickly socioeconomic issues. There are some high risk, if you like, communities within the population, and the reason that those are high risk are for cultural reasons, for language reasons. These are statistical relationships of risk. There's no implication of elevated levels of wrongdoing by any sectors of the community, but an effective program of outreach to those communities requires a sufficient level of translation of native language speakers who can speak directly to those communities and help them understand the problem. And probably an insufficient level of resource has gone into that issue so far in order to make that kind of program truly effective. So I'll stop there, but I hope I've illustrated that there are a very wide range of disease and vector issues that are intimately tied up with how successful the control program is likely to be, many of which have, at least in theory, policy level solutions through funding.
Okay, thank you. Any questions? Okay. You've given us a lot of food for thought and a lot to think about moving forward. So I appreciate your testimony very, very much, and look forward to working with you on some possible solutions as we move forward.
Yep.
Okay,
Thank you.
Thank you.
And at this time, we'll go ahead and take any public testimony or comments. Seeing none, we'll bring the hearing to a closure. Thank you very much.